Hello, very good morning. Children, today I will start the new topic, and in this topic I will cover the important words, important methodology of the food preservation. So the topic is the methods of food preservation. In this topic, as you know that the food is very important for our important component for our life. So preserving food is also very important, right? So when we start the contribution of the microorganisms, at the same time we should also remember the bad effects of the microorganisms. There are microorganisms which are very beneficial for our life, since we are making the medicine, we are preparing the antibiotics, we are preparing the antigen antibodies from the microorganisms. But at the same time, there are some harmful microorganisms which are causing bad effect in our life. Like you have heard that the food spoilage is the biggest problem for our countrymen. The food spoilage can cause bad effect in our physiology, in our health, in our body. So when we start this, the methods of food preservation, how we can preserve our food for the longer duration. We must notice that what are the methods by which we can easily preserve our food material without spoilage of food material. If there will not be any preservation, the loss of food items would be there. So for preserving food, what steps, what methods we are going to follow, I will explain those methods in this module, in this lecture, right? Before beginning this topic, the methods of food preservation, many of you have heard about the different popular methods. Like when you enjoy any pickles, when you enjoy any sauce, like in the market there are many items are available. So what things are present in that eatables, because of what things those eatables are can be used for longer duration. Must be there would be a certain chemical preservative. I'm sure that many of you have heard about that. Because of the preservative, they can keep that food items for longer duration, no problem. So I will explain about the different food preservative, different methods of the food preservation, by cooling them, by heating them, by mixing the salt, by mixing different materials. We can use the different food items, eatables for longer duration, right? So when we start the food preservation, I am going to write the few methods in front of you. Number one, the first one is chemical method. What we do in this chemical method, that in this method we always add some chemical preservative for keeping our eatables fresh, for making our eatables for longer duration usable. We should add some chemical. As it is available in the market, we will purchase these preservative, and by mixing these preservative we can save our eatables. Like in the pickles, in the sauce, in many eatables you have seen some composition, you can read that there are this chemical preservative is mixed. Like the example is sodium benzoate. There are many chemicals available in the market which are used for preserving the food items. By mixing these, sodium benzoate and other preservatives are also there. We can make our eatables fresh, free from the spoilage, free from the attack of the microorganisms. The second one is, I am going to tell you a very important method. In this method, you will see one theory on the basis of which it is working there. The by common salt, by mixing, it was number one by chemical, number two by common salt. It is very cheap and very easy. It is very clever thinking that when we are mixing the salt, how it can preserve that eatables, how it can preserve that pickles. In the orthodox method, your senior people of the house, always dadima, they mix the salt in the pickles. And what happened, you will surprised to know that in this common salt, one process would be taking place. Osmosis. What I am going to explain, what thing happened, by what the common salt preserve the food. Many of you have heard osmosis and diffusion. It is a process in which molecules move from its higher level to its lower level, like when we burn an agarbatti. So smoke is spreading from higher region to the lower, right? Got my point? Because of the concentration difference, it moves from one place to another place. When the movement takes place through the membrane, there would be end osmosis. Like when we put a dry kismis in water, dry kismis resin will swell. Why it will swell? Because it will absorb the water, right? So it will swell, it will absorb the water. And here, in the exosmosis, when we are keeping salt outside, what it will be there, the exosmosis will be taking place. Since the salt is hypertonic, having more concentration, try to understand this, then you will be able to write. This will absorb the water, right? The movement of water will be taking place outside. The exit of water, the thing, like I have kept the pickles. If there is moisture, if there is water is present in that, which is the cause of the infection of microbes, so that water will be drained out. Suppose you are putting a pinch of salt here. What it will do? It will absorb the water. Okay, it will absorb. So when we are keeping salt outside, when we are mixing salt, it will absorb the water from that very eatables, from that very pickles, from that very sauce or pickles. So there would not be moisture or water in that very pickles. So our pickle will be free from water by adding these very salt. Got my point? When we are mixing the salt, the exosmosis process takes place. Look at this. Through the exosmosis, it will drain the water outside, it will take the water, it will absorb the water from that very pickles, and the pickle would be free from the water. By this, you can make. It's a very easy, simple methods, orthodox, very old methods. Why they are mixing the salt, right? So it was the mixing of common salt. On the same theory, you can easily, when we are willing to save our pickles from the moisture for keeping longer duration, either we should add salt or we should add oil, or one method is also preservation by sugar. We can mix sugar. By mixing sugar, there would be osmosis, and by this we can also preserve our eatables for longer duration, right? Then by oil or vinegar, when we mix oil or vinegar, same thing would be happen. So by mixing all these chemical preservative, by mixing common salt, by mixing sugar, oil and vinegar, we can preserve, right? And also nowadays you have heard the many packing is done by the one vacuum is created, moisture has been taken outside, the eatables would be remain dry, and there would not be any attack of the microorganisms. That is also tanning and packing method. When vacuum is created, the moisture is taken out. So nowadays there are many latest and recent technology are there through which we can preserve the eatables, right? One is by heating and cold treatment. Number 4, by heat and all these methods are for the killing the microbes present in the eatables. Now coming to the heat and cold treatment, some when we are heating anything, and I think most of the packets of the milk, every packets of the milk you have heard pasteurized. I am coming to the, because this is very very important, that by heating and cooling, when we keep any eatables in the fridge, the enzymatic action would be reduced and there would not be any attack of the microbes, because in the low temperature microbes are less active and there would not be any bad effect of the attack of the microbes. There would not be any chances, because the preservation methods, we are keeping the temperature below 5 in the fridge, in the refrigerator, by which we can keep that. It is very simple and easy. Okay. Now moving to the heat. When we give heat, what we do and what is the name of that very preservation? I am going to write the example is pasteurization. In the pasteurized, I think you have heard the pasteurized milk. What we do in the pasteurization methods, we are keeping anything in a milk at 70 degrees Celsius temperature for the duration of 15 to 30 seconds. We are not going to heat in the 100 degrees Celsius temperature. We are going to heat at the temperature 70 degrees Celsius temperature and only for the 15 to 30 seconds. In this temperature, the microorganisms, the microbes, the bacteria which are present in the milk, that would be killed, and after killing that microbes, we will chill it, and in this method the temperature, how much temperature only required? Temperature 70 degrees Celsius for 15 to 30 seconds. Got my point? For this duration, for shorter duration, we are providing 70 degrees Celsius temperature. In that temperature, the microbes would be killed, and by this method, every packed milk, nowadays the packed milk is available, and by which method we are going to preserve that? By the pasteurization. So, in the pasteurization method, we are providing temperature, milk is heated at the temperature 70 degrees Celsius for shorter duration for only 15 to 30 seconds. This is called heat treatment and this is known as pasteurization. Got my point? So these are 4-5 methods by which you can preserve your food very well, right? So in the microorganism chapter, it's a last lecture for our boys, that in this lecture I have taken 3 modules, and in these 3 modules you have to complete the chapter. And before completing this last module, one topic is left, and already you have heard about this nitrogen cycle, but I will explain also that in the nitrogen cycle one very important things are there. One important bacteria is involved in the nitrogen cycle, which is fixing the atmospheric nitrogen in the soil. Can you name that? Yes. That is very good example of the mutualism. That is very good example of the symbiosis. One special bacteria which is present in the pulse family root, and the name of that bacteria, can you guess? The nitrogen fixing bacteria, and that is known as the nitrogen fixing bacteria, and the name of that bacteria is rhizobium. Can you remember? So it is playing a very important role in nitrogen fixation in the soil. This bacteria is always present in the pulses root nodules, and this bacteria fixing the atmospheric nitrogen in the soil, making our soil very much fertile because of one special gene in this bacteria, and that gene is NIF gene, nitrogen fixing gene. So moving to the last segment, nitrogen fixation on N2 cycle. Okay, so I am going to start. Nitrogen is present. What is the percentage of nitrogen in the atmosphere? It is 78%. But until unless this nitrogen is not going to fix in the soil, it is not usable form. When it is not going to be converted into usable form, this 78% nitrogen cannot be used by plants. So it should be converted into nitrite and then nitrate. So only the nitrate is the usable form, like sulfur usable form is sulfate. Same way, the nitrogen can be used in the form of nitrate only. So it is very important to convert into the nitrate, and for that there are many steps. 2-3 steps are required. So I would like to begin this nitrogen cycle, N2 cycle, like first is rhizobium bacteria, nitrogen fixation. Naturally, nitrogen is fixed in the soil by this, and then this is converted into ammonia by the ammonification. The nitrogen is converted into ammonia, and then this ammonia is converted into nitrite, and this nitrite is converted into nitrate. So you can write nitrification. So number 1, number 2, number 3. Then by the denitrification, it is returned back to the atmosphere. So there are 4 steps through which you can easily complete the nitrogen cycle. You can write the number 1, nitrogen fixation by the rhizobium bacteria. The rhizobium bacteria is fixing the atmospheric nitrogen in the soil. There are other method also through lightning. The few percentage of nitrogen is also going to fix in the soil, it reaching in the soil. But here by the this microorganism, by this rhizobium bacteria, it is fixed in the soil. Then that protein is converted into ammonia into the by the ammonification process. Nitrogen is converted into ammonia in the ammonification process, you can write. And this ammonia is converted into two more steps. Very first, ammonia is converted into nitrite by the nitrosomonas, and then that nitrite is converted into nitrate in the by the nitrification. So what is the nitrification? The conversion of ammonia into the nitrite and nitrate. It is called the nitrification. You got my point? The process of conversion of ammonia into nitrite and nitrate is called nitrification. Here the nitrogen fixation, the atmospheric nitrogen is fixed in the soil in the nitrogen fixation, but ammonification, protein is converted into ammonia is known as ammonification. What is the nitrification? This ammonia is converted into nitrite and nitrate is known as nitrification. And then this nitrate is usable. So nitrate is the usable form of nitrogen. Got my point? And this nitrate then return back to the atmosphere. You can see the arrow. Look at this, children of student class 8. They can easily understand nitrogen fixation, ammonification, nitrate and denitrification. By this, the nitrate is returned back to the atmosphere. So with the help of 4 steps, we can complete the nitrogen cycle. It's a very easy way to understand this. So denitrification, by the involvement of one special bacteria, you can write the denitrification process by Pseudomonas bacteria. By Pseudomonas bacteria, the nitrate is returned back to the atmosphere. So there are involvement of the 2 bacteria, especially rhizobium bacteria and number 2 Pseudomonas, which return back to the atmosphere. So this nitrate is returned back to the atmosphere with the help of denitrifying bacteria, and this denitrifying bacteria is known as Pseudomonas. The name of the denitrifying bacteria is Pseudomonas. So first nitrogen fixation is done by the rhizobium bacteria. The second step, the last step of the nitrogen cycle, the denitrifying bacteria, the Pseudomonas, is returning back, is taking this nitrate, taking back to the atmosphere. So with the help of these 4 steps, you can easily complete your nitrogen cycle. So in today's lecture, you have completed the methods of the food preservation. And the second one, you have completed the nitrogen cycle, which can be asked. There would be question that how you can make the soil fertile by involvement of the rhizobium bacteria. You can make soil or soil fertile. Okay, there are important uses of the microbes in our daily life, and the nitrogen cycle is completed. The food preservation methods are also completed. In this lecture, we have completed the 2 topics, and before this I already taken the uses of the bacteria, the bad effect of the bacteria, like the table you have to prepare, the bacterial disease table, viral disease table, and then microorganisms, types of microorganisms, protozoa, algae, fungi, bacteria and viruses. So this is the microorganism chapter. In this way we have completed the microorganism chapter with the help of these 3 modules. I am sure that you will be able to write the question answer, fill in the blanks very well in this. With these contents, you can easily write your answer, questions answer, right? So before ending the module, conclusion of my lecture, I would like to give you some assignment questions. mention the methods of food preservation. Mention any right? 4 methods of food preservation. Then moving to the next one, what is pasteurization? As I explained that heating milk at 70 degrees Celsius temperature for the 15 to 30 seconds only, by this we are going to kill the bacteria and we will make our milk free from the bacteria. And you know, every day we are using the pasteurized milk in this method. So it is by the heating any eatable milk is known as the pasteurization process, right? So number 3, you can explain the nitrogen cycle. Explain the nitrogen cycle. The last question I would like to again mention that explain the role of bacteria in increasing you have to explain how the bacteria is increasing the soil fertility, and I'm sure that you are confirm the role of bacteria. I already explained in the crop rotation also, when you grow the pulse family crop, what special things happen. There are nitrogen fixing bacteria. Rhizobium is present in the root nodules, and by the nitrogen fixation in the soil we can increase our soil by mixing the atmospheric nitrogen in the soil. And when the soil would be rich of this nitrogen, they will show the flowering, fruiting very fast. So for increasing the rate of flowering, fruiting, germination, the very important mineral is required, and the important mineral is the nitrogen for increasing the flowering, fruiting, germination in the plant, in the crop. So these are the 4 questions for the assignment, and the first one is, I am sure that 4 methods: by mixing salt, by mixing vinegar, by mixing chemical like sodium benzoate. You can easily write the answer of the first question. The question number 2, what is pasteurization, is not tough. You can write it is the method of heating milk at the 70 degrees Celsius temperature for 15 to 30 seconds, right? And the nitrogen cycle, which consists of 4 steps: nitrogen fixation, ammonification, the third one is nitrification, and the fourth one is denitrification. By these 4 steps you can easily complete your nitrogen cycle. Okay, and the last, which is already I explained you, the role of bacteria in the soil fertility. So when you complete any module, any lecture, and now it's your duty, it's your responsibility to complete the assignment questions. I'm sure that you will be writing these questions for today's module, right? So I would be meeting with my new contents, with my new topic in the next day, and till then, bye bye, take care.